Mendel was a great guy, but sometimes traits are not simply binary choices but can exist along a gradient. In this lecture, we're going to cover quantitative genetics and QTLs, quantitative trait loci. Let's get rolling. I'd like you to be able to contrast between Mendelian and quantitative genetics, define QTL and LOD score, be able to relate LOD scores to linkage and even to selective sweeps, relate genotype and environment, and explain when to use a common garden. Define and explain how H² is used and compare Fischer and Orr's hypotheses on QTL impacts. So let's start with some quantitative traits. So there are traits that are qualitative, red versus white flowers, and there are some traits that are quantitative, such as height. Not only that, but I'm sure you know that height is going to be environmentally impacted as well, thus complicating any measure of inheritance. We could look at the height measures in pea plants of Mendel's peas and see that they are either tall or dwarf, but then when we look at a whole group of humans, we see that they are neither just tall nor dwarf, but there is a whole gradient of heights. And not only that, but it seems to be somewhat gender specific, with the women here dressed in white and the men in black, having different heights based on their chromosomes. So we've got a lot to deal with genetically that is not specifically Mendelian. So there is a Mendelian approach to genetics, and that is a simple inheritance of either or, where an allele is either recessive or dominant. And I say an allele, as in one. So if there is one, we might see this, dominant or recessive. But what about codominance? Well, that's non-Mendelian already. So we get codominance where the intermediate, the hybrid, if you would, is going to have a higher height than not. So here we have a short. This is a Nicotiana species, tobacco. You have short flowers and tall flowers. They have just longer sides and they're a little bit bigger. So you mate them, and the intermediate is intermediate. So what we see there is when the intermediate, the F1, is allowed to self-pollinate, you get three different heights: short, medium, and tall. However, if there were two loci, what you're going to get is short, tall, and three different types in between. Could they 9 to 3 to 3 to 1 approach, or specifically a 9 to 3 to 3 to 1 kind of approach between these different alleles being parsed out. And then you have, if there were 6 loci, well, that's even more. And we see is as you increase the number of genes that are responsible for a trait, you are going to increase the number of genetic combinations that will come out. I know in genetics you did a simple cross with a 3 to 1 and a dihybrid cross with a 9 to 3 to 3 to 1, and here you see some nightmares with a 6-way cross. Mwahaha, let's not get into the math, let's get into different types of math. And this is when there are going to be greater than 2 loci. Each one of these loci is going to be called something called, and so it produces a messy graph. So you start with these short ones, and the short ones aren't just one size. It's not just one corolla length. The tall ones don't have just one corolla length. They're actually a little around a certain corolla length with variation in just a few millimeters. If you cross them, you get all intermediates, but again, variations around a central thing with, a few millimeters. The F2 generation has a greater amount of variation, but still doesn't bring back any of the the parental traits. So you don't have any super short or super long. However, after generations of artificial selection for short corollas or long corollas, you can get them back into 2 camps. It's not as clean. It's quite messy, actually. But if you continue that selection, you could probably parse them out even farther until you have restored the original parental lineages. But you can see how within 3 generations you still haven't selected all the genes, back in and back out like with a simple Mendelian cross. And that's because these are quantitative trait loci, or QTLs. There is a locus that has a quantitative amount of length that it will give a corolla. And that quantitative trait locus could be just one gene, or it could actually be a couple genes acting in tandem, perhaps even under the same transcription factors and under the same promoters. Ooh, spooky. So disentangling these is complicated. So how can we do this? Well, let's go through an example of QTLs where we can see different species being kind of mapped out. So we have here Mimulus cardinalis, which is a red species of Mimulus monkey flower that is going to be pollinated by hummingbirds. Hummingbirds love red, has long corollas. Okay, then you get Mimulus lewisii, shorter corollas, and it has bee pollination. It's a yellow flower, and they're enjoyed by different species. So the question here is, what are the genes that lead to flower color and shape? And one of the assumptions is that these would be discrete, so that each gene would have a very specific effect on flower color, or a very specific effect on corolla length. So we can see discrete units of flower shape and color being mapped out at the different loci. So each one of these is going to map to a different part of the genome. And there are two models of how these QTLs could be mapping out. And one of them is by Fischer. And what he said is that alleles give small effects and are driven to fixation. So that we have 50 short steps between Mimulus lewisii and Mimulus cardinalis. Whereas Orr would say, no, you've got one leap, 5 jumps, 10 paces, and then 34 small steps. So it could still be 50 genes, 50 loci responsible, but they have very different effects, such that you get very close early on. Think about that paradox. I can't remember the name of it. Basically, you always get halfway. Zeno's paradox. So that would be closer to what Orr would be saying. But he would really say that alleles are fixed. They have large effects first, and small effects maybe later as you're kind of optimizing. Whereas Fischer would say basically all alleles are kind of the same. And that can be true with Darwinian evolution, where you have the very slow gradation between two different types. So here we go. Oh, that was, by the way, these are pictures of all the different hybrids possible between Mimulus lewisii up in the upper corner and Mimulus cardinalis in the lowermost corner. So you can map these, though. So you can map loci. What you do is you find something called a marker. So this is going to assume that you already have things you know about the species. So like for tomatoes, for example, you have a trait where the tomato plant is going to have an exserted pistil. So the little pistil comes out of the pollen tube. That's a trait. You can see that trait. If you know where that trait is on chromosome 3, then you can say, okay, this trait is on chromosome 3, and if my trait for smaller fruit also maps alongside the exserted pistils, so if a plant with smaller fruit always has exserted pistils, these are two traits that are probably linked and probably both on chromosome 3. But if you're just looking at, okay, I have smaller fruit and I have exerted pistils, or I have larger fruit and I have a genetic sequence that I know is variable. So you have some sort of area that you have mapped out using PCR, put on a gel, and you see they have different lines, so you know that this is where it is on certain chromosomes. So if you know where the markers are on the certain chromosomes, that's tricky getting that first, then you can say, okay, does my trait go with the chromosome? And if it does, you've got something called linkage. If your trait is genetically linked to the marker, what you would see is you'd have your two parents, one of whom has small fruit and exerted pistils, and the other which has large fruit and this genetic sequence that we can know would be heterozygotic, or either one of homozygotic. It has to be, I guess, homozygotic long sequence. There we go. And you mate them. Your F1s are going to have, all the traits, but you have your F2s. Do those things segregate together? If they segregate together, then your marker is linked to your target. If they do not segregate together, then your marker is not linked to your target. So if you get plants that have exerted pistils and large fruit, at the same rate that you get plants that have exerted pistils and small fruit, that means it's basically that 50-50, they're not linked. They might even be on separate chromosomes. So you're checking to see if a trait and a marker are linked by looking at the F2 generation. Might be a good time to review what you know about linkage or request that I make some sort of video about linkage if you need it. I can go over super basic genetics, I can go over linkage. All right, so you map those QTLs, and what you're going to then go for is what's called an LOD score. Math. It's a logarithm of the odds. So you're checking to see what is the likelihood that a marker and locus would co-segregate compared to the assumption of each of them just kind of existing on their own. And if they are linked more often than you would expect, then they have a higher LOD score. Now the LOD score, of course, has to be above a certain threshold. You set the threshold for you to say this marker and this trait are linked. So let's say there's a selective sweep. So here we have pH. So having a tomato, again, of a certain pH changes its marketability. So you're looking for what alleles, what loci give the pH trait. And you look on chromosome 10, you've got a whole bunch of markers on chromosome 10, and you see which markers co-segregate with pH. And you see that you have an area on chromosome 10 where these markers are co-segregating with the pH trait more often than not, and more often than an LOD score of 2.2 here. So that means that the pH is probably between these markers where it crosses 2.2 and where it goes back down below 2.2. Now you can set the LOD score higher, but when you do that, you risk that there might actually be the marker co-segregating for other reasons. So it's tricky to really map it down closer and closer, and it requires more and more introgression lines where you're crossing it back to the high pH or low pH line and seeing where this trait segregates based on the markers and separating the markers. Remember, if something is very close genetically to another locus on a chromosome, they are going to co-segregate until recombination breaks them apart. And recombination of something that is 3 centimorgans away only happens 3% of the time. So you really need a lot of offspring, a lot of back crosses to really get a good LOD score and narrow down the area where your trait is, where your locus is. And there are multiple loci, perhaps on multiple chromosomes, that you will be tracking through this method. QTLs are hard to find, or you can get lucky. So we can talk about that in a minute. But this Fischer or model. So this is your this is actually Shemsky and Bradshaw 2002 is this paper. Cat. They were checking this Mimulus cardinalis versus Mimulus lewisii, and they crossed them, and they back crossed them, and they back crossed them again, and they found a lot of the QTLs have less than 20% of the variance explained. Okay. So when you have less than 20% of the variance explained, that is small effects. But they also found some genes of very large effects, including one gene, one locus, where the pollinators actually switched. So you take a wild-type Mimulus lewisii, which is bee pollinated, and you add the locus, you actually get it pollinated by pollinated by hummingbirds, just like that. So one locus can cause a switch of pollinators. So this goes much more with Fischer's idea. Orr's idea. Orr's idea, where you have these, go back here, alleles fixed have large effects. So that one allele that can switch between hummingbird and bee, that is a large effect. After that, it's more small alleles that change the corolla shape, that change the presentation, where the pistil is, where the stamens are. All these are smaller, later effects that do increase the fitness, but only have 20% or less of the variance explained. So we see this one actually comes out more like Orr. Now there are different examples where you'll actually see a Fischer's model work well. So these are not necessarily right and wrong, it's right or wrong for each example. Now sometimes you can get lucky in finding candidate loci. So when what you're doing here is you look at a marker, and the marker might have a certain thing that you might expect with the trait. So you check to see if there are any genes in your known region. So once you've narrowed down that LOD score sufficiently, you see, are there genes in the region that we know about? So here we have a gene like, oh, it's for this gene causes a difference in a proton pump. Ah, could that maybe work with acidity? Let's find out. Maybe it does. Good job. You found the gene responsible for pH in tomatoes. Nice. It's actually worth something if you know what you're looking for. If you are just looking for areas that look gene-like, you might actually accidentally pick a pseudogene. Oops. Or if you have areas that have the potential traits that could result in your target trait. Here we go. They checked out, they found a bunch of rats and they looked for ones that were more novelty-seeking. So they mapped it, of course, out to a certain region of the genome, and they knew that in that region of the genome, there was a dopamine receptor that they were using kind of as a marker. And they found that this dopamine receptor and the novelty-seeking score really correlated well with each other. In fact, to the point where those rats that had this specific dopamine receptor were also having a higher novelty seeking. So it is very likely that the novelty seeking trait is due to a change at the alleles of this dopamine receptor. And that's what you call candidate loci. And that's kind of the goal of a QTL search. All right, take a deep breath. We're going to move into heritability. You might want to pause it now. I do want to put these together. Heritability. So we know variability is necessary. Heritability is necessary. And heritability can be, of course, measured. So QTLs provides the tools we need to measure how much, what quantity something is inherited. But we need to disentangle nature and nurture. This is where identical twin studies are just perfect because they have the same genes. We can see what we can do to change them. Here's Mark Kelly and Scott Kelly, one of which was sent up to space, one of whom was staying on the Earth for a long period of time. We can see what changes the environment of space had on their physiology, knowing that we have the same genes. All right, so there is here broad sense heritability. And broad sense heritability is looking at the fraction of the total variation due to genes versus due to the environment. The total variation is your phenotypic variation, and that is your, remember your reaction norms, like how dark a caterpillar is, whether a gecko is going to lay mostly male or mostly female eggs. That's your reaction norm. And you can measure this broad sense heritability as variation in genes divided by the variation in the phenotype. That's your broad sense heritability. Well, what's variation in the phenotype? Variation in the phenotype can be measured by variation in the genes that we know of and variation we know of in the environment. So it's disentangling the environmental from the genetic and seeing how much is actually heritable when you actually start pulling this apart. This number is always between 0, which is only in the environment determines things, and 1, which is only genetic. So like my hair length is going to be an environmental factor. It determines how often I'm in encounters with the clippers. My eye color is going to be only a genetic thing, unless I'm wearing contacts, special contacts. But this is broad sense heritability, is the degree of genetic determination given in a certain environment. Then we have narrow sense heritability. This is much more quantitative. So narrow sense heritability requires that you do some crosses. You get a whole bunch of parents, and they have to vary based on some certain trait. Now you don't just make the tall with the small and the small with the tall, or everything's going to be average. A lesson I should have learned before doing my own one of these. You make tall with tall, small with small, tall with small, medium with medium, all the different variables, and you get the mid-parent and the mid-offspring values. So the mid-parent is you take the two parents, you measure their trait, and you average it, and that's the mid-parent value. For the offspring, you take all the offspring that came from that cross, and you measure all of their traits. So measuring all of their traits is going to give you your mid-offspring value, so the average of all the offspring. So how well does the mid-parent value on the x-axis correlate with the mid-offspring value on the y-axis? And more importantly, what's the slope? So the slope of the line is something called h squared. So you just put a correlation, do a linear regression, and find the slope of the line. H squared is your narrow sense heritability. It's how much the parents' values are going to change and in which direction the offspring's values. What you also have, for fun, here is your r squared value. your R squared value is how much of the variation in offspring is explained by the heritability and variation in the parents. So that's kind of a cool other thing you can see is really how much of this is, not due to environmental factors. So if it's strictly genetic, an R squared value will just be 1. But if it's strictly environmental, an R squared value will be 0, being very similar to how we're measuring the broad sense heritability. Then we have additive and dominance variation. Additive is when multiple genes are going to have a cumulative effect. Think back to the QTLs. Think also in codominance, where two alleles are going to have some sort of an additive effect to create an intermediate phenotype. Then there's dominance variation, which is due to certain genes being dominant or others. So you can make this whole equation where H squared equals the variation in the additive, variation due to additive, divided by the variation due to phenotypes. Okay. And then you're going to take that and make it into variation over additive is divided by the variation of additive, variation in dominance, plus variation in the environment. Here's just another much more complicated way of measuring narrow sense heritability. Personally, I just recommend doing the crosses and measuring all of the offspring. It's actually a little bit easier. So that is our objectives 1. That's a lot of material. You're probably going to want to go back through this. I really recommend reading the book, doing the homework, of course. Check through these to see how well you understand the objectives. It's a lengthy lecture. You might want to watch it twice.